Hello everyone, welcome to today's lecture. In this lecture, we will build on what we have learned in the previous four sessions and learn how to actually culture microorganisms. Culturing microorganisms is an important technique in microbiology, and we can study their growth, characteristics, and metabolic activities. This is what we will cover today. In last chapter, we classified culture media based on their physical state into liquid, solid, and semi-solid media and studied their characteristics. The choice of culture method depends on the type of medium used. Today, we will learn about liquid culture method using liquid media and solid culture method using solid media. Additionally, we will discuss colony counting after streaking on solid media and introduce the CFU colony forming unit method for estimating viable cell numbers. Microbial culture can be divided into liquid culture and solid culture. Liquid culture is commonly used for growing microorganisms with different methods. Aerobic bacteria are typically cultured using shaking incubator, while anaerobic bacteria can be cultured using soft agar, though specialized equipment may be required for strict anaerobic conditions. The general procedure for liquid culture begins by dispensing 4 milliliters of TSP medium into sterile test tubes. A prepared microbial sample, such as Salmonella, is then inoculated into one of the test tubes at a volume of 100 microliter, followed by voltaxing to ensure proper mixing. The inoculated test tubes are placed in a shaking incubator and cultured at 37 degrees Celsius for 24 hours. Solid culture is primarily performed using streaking and spreading method, with the choice of technique depending on the experimental objectives. The pour plate culture is another method that mixes microorganisms within the medium itself. The images below show actual results from solid culture experiments. In the streak plate method, an inoculation loop is used to streak bacteria across the agar surface to obtain isolated colonies. This method is useful for storing pure bacterial colonies for up to 3 to 4 months. The streaking process involves making 3 or 4 streak patterns, gradually diluting the bacterial concentration. Initially, a high concentration of bacteria is streaked onto one part of the agar, but with each subsequent streak, the bacterial concentration decreases, resulting in individual colonies forming in the final streak section. Make sure to sterilize the inoculation loop between each streaking. In the spreading method, a microbial sample is evenly spread across the agar surface. A prepared sample is pipetted onto the agar plate. Using a sterilized spreader, the sample is then evenly distributed across the surface by moving the spreader back and forth. This process ensures uniform distribution of the sample, allowing for optimal microbial growth. The pour plate method is another solid culture technique, where diluted microbial samples are mixed with liquid agar before solidification. The process begins by pipetting a diluted sample into a petri dish. A sufficient volume of liquid state of medium is then poured into the dish and mixed gently to even distribution. The medium is solidified at room temperature. After incubation, colonies form both on the surface and within the agar. Strip plate massive, the back of an agar petri dish is labeled with the media type, sample name, date, and dilution factor. An inoculation loop is flame sterilized and cooled before being dipped into the microbial sample. The sample is streaked across one quadrant of the agar surface using the loop. The loop is then flame sterilized again, and once cooled, the loop is used to streak from the first quadrant into second quadrant, gradually diluting the sample like the picture on the right. This process is repeated for the third and fourth quadrants. As streaking progresses, the bacterial concentration decreases, and isolated single colonies can be obtained in the final section. After streaking, the TSA plate is incubated at 37 degrees Celsius for 24 hours. In the spread plate method, the back of an agar petri dish is labeled with the media type, sample name, date, and dilution factor. The sample is diluted to 10 to the minus 5 using PBS. A sterilized spreader is prepared by dipping it in alcohol, flaming it, and the spreader is cooled. 100 microliter of diluted sample is then pipetted onto the agar surface. The sterilized spreader is used to evenly distribute the sample across the plate. Once spreading is complete, the spreader is sterilized again to prevent contamination. The TSA plate is then incubated at 37 degrees Celsius for 24 hours. In the pour plate massive, the back of an agar petri dish is labeled with the media type, sample name, date, and dilution factor. A pipette is used to transfer 100 microliters of a 10 to minus 5 diluted sample into the center of sterile petri dish. Next, sufficient volume of liquid TSA medium is carefully poured into the dish. The sample and the medium are mixed gently to ensure even distribution. The TSA medium is solidified at room temperature. After TSA plate is incubated at 37 degrees Celsius for 24 hours. The forming unit, CFU massive, is used to estimate the number of viable cells in a sample by counting the colonies formed on the medium. CFU is expressed as CFU per milliliter, representing the number of viable cells per milliliter of culture. The procedure for CFU involves serial dilution and spread plate massive and incubating the plates to count colonies. If the sample is too concentrated, excessive colony formation occurs, making it difficult to count individual colonies. To obtain reliable results, serial dilution is essential. The optimal number of colonies for CFU counting is between 30 and 300. The CFU value is calculated using the following formula. Count the number of colonies on the plate and divide it by the total dilution factor. For the past 4 lectures, we have covered the fundamental techniques for microbial culture. In Chapter 1, we explored the importance of sterilization and its method. Chapter 2 introduced concentration calculation and buffer solutions. Chapter 3 showed different types of culture media and their characteristics. And Chapter 4 focused on culturing techniques and streaking method. I hope this information has been useful and helps you with culturing microorganisms in the lab. Thank you for your attention.